Hello everyone, let us continue with the chapter Molecular Basis of Inheritance Part 15. We are going to continue with a new topic. We call that to be operon. So in prokaryotes, that is in E. coli, we are going to learn a new topic that is called as operon, operon concept. So what is an operon? A set of genes that is controlled by a promoter is called as operon. A set of genes that is controlled by a promoter is called as operon. Okay, so what is that example? If the lactose is used as a medium, you call that to be what? Lac Operon. If tryptophan is used as a medium, then you call it as Operon. If arabinose is used as a medium, then you call it as ARA, arabinose. Okay, then valine, histidine, operon. So here you call that to be lactose, then tryptophan, arabinose, and then valine, and his stands for histidine. Okay. Now here we are going to study in prokaryotes. In prokaryotes, what is prokaryotes? Very good example, E. coli bacteria, E. coli, Escherichia coli. Right? So in E. coli, we are going to learn about a new concept that is called as operon. So very good example, what you are going to learn here is about Lac Operon. So what is operon? A set of genes that is controlled by a promoter is called as what? Operon. Eukaryotes. Eukaryotes, what did we learn? We have learned that in the transcription unit, if this is the transcription unit, 3' to 5' is the template strand and 5' to 3' is the coding strand, and we have this promoter, right, and the terminator. So in between the promoter and the terminator, you call this as structural genes. What do you call this as? Structural genes. Where it is present? In between. So this is the promoter and this is the terminator. So in between the promoter and the terminator, structural genes are present. Okay. But in eukaryote, you call these structural genes, if the structural gene that is present in a transcription unit, okay, in eukaryote, it is said to be monocistronic. Monocistronic. Then what about in prokaryotes? In prokaryotes, the structural genes that is present in the transcription unit, it is said to be polycistronic. So you can very well remember PP, prokaryotes and polycistronic. So in prokaryotes, it is poly. Poly means many cistrons are present, many structural genes are present. Whereas here in eukaryotes, it is monocistronic. There are two types of operon. One is called as inducible operon. Inducible operon. The other one is called as suppressible operon. So what is inducible operon? Here, under the normal condition, the operon will be kept on, whereas if it is induced, it will be kept off. So what is the example? Here, Lac Operon, it comes under inducible operon. Then what happens here? Under normal condition, it will be off, but when it is induced, it will be on. So again, what is the example here? Tryptophan operon. Understood? So here, we have learned that under normal condition is on and induced condition it is kept off. So what is that? When lactose is present, here in this prokaryotes, we are going to concentrate on Lac Operon concept. We are going to concentrate only on what? Lac Operon concept. Okay. So when you study about the Lac Operon, if lactose is present, whether the unit is kept off or on, or if it is absent, what happens? That is the main concept what you are going to learn here in this prokaryotes. So here we have learned that structural genes are present. So what are those genes? In this Lac Operon, we have three here: Z gene, Y gene and A gene. Z gene, Y gene and A gene. And we have learnt in transcription unit, you have a promoter, right? But here, there is an operator. Okay? And here, you have this promoter, right? And one more difference what we are going to learn here in this Lac Operon unit, one more gene is present. It is an I gene. And one more promoter is also there. So in Lac Operon, you have 2 promoters and 1 operator. So what happens here? If the transcription have to take place, or if the gene, if it has to function, first, what did you learn in central dogma of protein synthesis? Transcription and then translation, so that a particular protein will be synthesized. So if the I gene is transcribed, what happens here? Transcription have to take place, followed by what? Translation. So if translation take place, protein will be synthesized, right? Protein will be synthesized here. So what is this protein called as? You call that to be repressor protein. What do you call that as? Repressor protein. Okay. Now, here I have told you about 3 genes. One is the Z, the other one is Y, the other one is A. Now what happens here? This Z stands for β-galactosidase. Beta-galactosidase. Okay. And Y codes for the enzyme called as permease. And then A codes for enzyme called as transacetylase. Transacetylase. So Z codes for an enzyme called as beta-galactosidase, Y codes for an enzyme called as permease, and A codes for an enzyme called as transacetylase. Okay. Now, what did we learn? Transcription. During transcription, the genes are being transcribed. The genes, what is present in DNA, right? The information what is present in DNA, it is transcribed to mRNA, which will be helpful for the protein synthesis. Right? Okay. So, if the transcription has to occur, what happens? We have learned that in transcription unit, if the RNA polymerase enzyme comes and binds to the promoter, transcription will start, initiation will start. That's what we have learned in the transcription unit. So this is the transcription unit, promoter and the terminator. Okay, so this will be the template strand and this is the coding strand, right? So the information present here, it will be copied to the mRNA. So this is what you have learned in transcription. The same way. So what happens here? If this process has to continue, what is that enzyme called as RNA polymerase enzyme have to bind to the promoter. What is that enzyme? RNA polymerase enzyme. Okay. So only if RNA polymerase enzyme comes and binds to this promoter, these genes can be transcribed. Transcription can take place. Okay. Now what happens? We have learned that if this has to continue, this I gene codes for a particular protein. Okay, if the protein have to be synthesized, what should take place here? Transcription, then translation, then protein synthesis take place. That protein only, you call that to be what? Repressor protein. Understood? Right. Now what will happen? What is the function of the beta-galactosidase? What happens here when the E. coli, when it is allowed to grow in a medium containing lactose? What is that lactose? It is a medium. So, the E. coli will not be able to uptake the lactose as it is, because it is a disaccharide. Lactose is a disaccharide. Right. So, what happens here? This lactose should be converted into galactose and glucose. Lactose should be converted to galactose and glucose. Right? Okay. So, how it can be converted? With the presence of an enzyme called as beta-galactosidase. So, what is the function of this beta-galactosidase? It converts the lactose into galactose and glucose so that it can uptake very easily. Okay. So, this I gene, you call that to be inhibitor gene. What is this I gene? Inhibitor gene. And then there is one more promoter. But there in transcription unit, we have studied only one promoter. But here in Lac Operon unit, we have 2 promoters. Right? So what happens here? First concept, the I gene, if it has to produce a protein, it has to be transcribed and then translation take place and then protein should be synthesized. This protein is called as what? Repressor protein. Is it clear? Okay. Now, in the absence of inducer, in the absence of inducer, what happens? Right? Okay, so what is this lactose? Lactose only acts as an inducer, right? So what is this lactose? It is an inducer. It acts as an inducer. So when there is no lactose, what happens? This repressor protein, it will go and bind to the operator. So if it goes and binds to the operator, RNA polymerase cannot come and bind to the promoter. Only if RNA polymerase binds to the promoter, transcription will take place. So if this RNA polymerase, if it does not bind to the promoter, transcription will not take place. Transcription will not take place. Okay, in that case, you call that to be switched off. What do you call it as? Switched off. Understood? Next, what happens in case if there is presence of inducer? So what happens here? You are going to add lactose. If the lactose has entered, so what did I tell you? Beta-galactosidase, it helps in converting the lactose into galactose and glucose. Right? So here, next enzyme, what is that? Permease. So what is this? Permeability. So this enzyme helps the lactose to move inside. So when lactose is present, if the lactose is present, what will happen? This repressor protein cannot go and bind to the operator when there is presence of inducer. So if it does not bind to the operator, this RNA polymerase, it can bind to the promoter so that transcription will take place. This condition, you call that to be switched on. So what happens here? mRNA will be synthesized. When it becomes inactive. Right? So this is what you have to understand. In the presence of inducer, what will happen? In the presence of inducer, the repressor protein will become inactive so that it will not bind to the operator. If it does not bind to the operator, the RNA polymerase will come and bind to the promoter. If it comes and bind to the promoter, the initiation will start. Transcription will take place so that all these genes can be transcribed. Okay, if transcription take place, translation will occur and the protein will be synthesized. But in case, if there is absence of lactose, this repressor protein will become active and it goes and bind to the operator so that RNA polymerase cannot bind to the promoter, so transcription will not take place. Only two things you have to remember. In the case of absence of inducer, transcription will not take place and the condition you call that to be what? Switched off. First step, in the absence of inducer, transcription will not take place and the condition you call that to be switched off. Right? But next step is, in the presence of inducer, transcription will take place and the condition you call that to be switched on. So if transcription take place, mRNA will be synthesized and it will be ready for protein synthesis. So this is what we learn about Lac Operon in prokaryotes. If you like this video, share, comment and subscribe my YouTube channel and follow me on Facebook and Instagram, and the link is available in the description box. Thank you.